All right. So everyone, I guess it's now's a good time to start. So welcome to the Melbourne Integrative Genomics webinar. I would just like, before I get to the introduction, to remind everyone to turn off their microphones and also their cameras to preserve bandwidth. It's my pleasure to introduce today Sebastian Duchene. Sebastian works at the Doherty Institute. So he got his PhD in 2015 from the University of Sydney, working together with Simon Ho. He has had a number of postdocs. So he was a postdoc at the University of Sydney with Edward Holmes to 2016. For a few years, he was a Mackenzie Fellow here at the University of Melbourne in the Old Centre for Systems Genomics. And since last he has been an ARC DECRA Fellow at the Doherty Institute. So Sebastian clearly works in computational biology. He has specific interests in phylogenomics and phylodynamics, in pathogens and infectious diseases. And since obviously there is one disease which everyone is thinking about right now, he is here to tell us about that. And so he's going to be talking about bridging the epidemiology evolution of SARS-CoV-2 using near real-time phylodynamics. So before I hand it over to Sebastian, I will also say that he will be after the seminar for questions, but then also after that for sort of a meet and greet with PhD students, to sort of all students. So all you need to do is just hang around on the channel after the seminar. So with that in mind, take it away, Sebastian.
Thank you so much, Yaban, for the introduction, and thanks to Andrew and David to give this talk. So I'm just going to turn off my video while I talk, just in case that helps. Right. So is the sound all right?
Yeah, I can hear it.
Okay, cool. So in this talk, I'll be presenting some of the analyses that have been possible using the large amount of genome sequence data that are available, kind of freely available today. And I'll also discuss some methods that we've been developing to improve our capacity for nowcasting. It is a bit, we're still at a benchmarking stage, but I think we have some quite promising results. So hopefully this is something that will help us with our outbreak here. And whoops. Yeah, the first thing I'll mention is that there's a few excellent resources online where we've been following, or you can follow analysis, data, and also share different preliminary results that you might have. So the first one is GISAID. This is, it stands for Global Initiative for Sharing All Influenza Data. And a lot of the coronavirus data has been deposited here. Some of it is in GenBank, but most of it will be in GISAID. And if you register on it, then you can download the genomes and use them. There are a few things to consider if you want to analyze the data and publish using them, but it's quite easy to access. The other interesting or useful tool is NextStrain. And NextStrain pretty much aggregates a lot of the sequence data from GISAID and from GenBank and conducts some exploratory analysis, such as a phylogenetic tree. It will give you information about where the samples were collected, when they were collected, and it also keeps track of some useful or potentially useful mutations and classification of the virus into different lineages. And the other one I find quite useful is Virological. So virological.org is a blog where people working on different viruses, not just this one, have been depositing preliminary analysis. So this is different to a preprint. If you do something that you find interesting, you can post it here, and you'll get feedback from many of the people doing some of the most notorious work. So it's very useful, and I think it is a potentially good way to start collaborations and make sure that the analysis that we conduct are relevant and just properly done. So keeping track of the data in GISAID is a bit tricky. At this point, there's around 13,000 genomes from human samples from different places in the world. And here, so the y-axis is the number of genomes in GISAID, and the x-axis is the time when the genomes were collected, not necessarily when they were deposited in GISAID. And these different lines correspond to different geographic regions, those that have contributed a large number of genomes. So the USA has deposited by far the most genomes, followed by the UK, Australia. Iceland, I put it here because Iceland is an interesting case. They have a very small population size, which means that keeping track of their outbreak is easier. So they have a small number of genomes, but with respect to the population size, it's actually quite big. And this is China here. So we've deposited a sizable number of genomes in GISAID. But when we look at this picture, by dividing the number of genomes by the number of confirmed cases, of course, this depends on how intensive the testing is, what we find is that Iceland kind of tops the chart. But then we have Australia. So we're in a very privileged position in the sense that we're doing a lot of testing, but we also have a lot of genomes. We're pretty much sequencing almost every confirmed case. So we have very dense, very good sampling. And it's followed by the UK, the USA, and China here. And then if we zoom in to the pattern in Australia, so for the different jurisdictions, this is all the genomes for Australia, and Victoria is actually at the top. So we're sequencing more than other states. And then if we divide that by the number of confirmed cases, then we have the highest sampling effort and sequencing effort in all of Australia. So I think we're in a very good position in Australia, but particularly in Victoria. And of course, this is a mixture of having a relatively small outbreak and the resources to do the sequencing. So we have one of the densest sample data sets out there. Now, from the data of GISAID, people have been doing very interesting analysis and depositing them in virological. This is a few titles out there. I've deposited a few things myself and gotten feedback from very knowledgeable virologists. Some of the analyses that have been deposited here include papers that have ultimately been published in important journals, such as this one in Nature. This one here is actually a paper that is looking at different conspiracy theories of the origin of SARS coronavirus and explaining why it is probably a natural occurring virus. So a lot of useful stuff here. And this particular one was deposited, I think, a couple of weeks before it actually came out in Nature. So that gave everyone the opportunity of commenting and looking at the evidence behind this article. So let's have a look at what NextStrain is telling us and other tools that have been developed thanks to NextStrain and GISAID. One that I find quite important is this one called Pangolin. So these authors proposed, and this again was in virological, a classification scheme for the virus. Typically what would happen is that we wait for the virus to evolve an amount of time and accumulate some genetic change. We might see different subtypes or strains appearing, but we're still at the earlier stages of this particular outbreak. So classifying the virus into subtypes or things like that is a bit trickier. So they've proposed a dynamic classification scheme where the virus is classified into different clades, so monophyletic groups. And they've classified them from A to B, and then within each of these clades, you can have subclades. Note that I ran this, I think it was yesterday. So sometimes the colors change, sometimes there's a few differences in the classification scheme as more genomes are generated, and that's why this tool is useful to improve the classification. All we have to do is give it a fast alignment, and then it will use a reference set of samples from the global diversity to tell us what lineages they belong to. And I should also highlight that this lineage classification scheme is similar to what we use in bacteria. So an important question is, well, what diversity of the virus do we have in Australia or in particular geographic regions? And what we have in Australia is actually a representative of the global diversity. So I've highlighted here the Australian samples across the tree. And what you can see is that our samples kind of span the whole. Can someone mute their... Okay, I think that works. So we have a representation of the major lineages in Australia. And again, this is a result of intensive sampling and the fact that most of our cases until recently were travel importations. This is the same picture for Victoria. So I've highlighted here different Australian states. And again, because we've sampled so much, we see most of the cases here are Victorian, but we do know that New South Wales has a bigger outbreak. They just haven't deposited as many sequences in Jezade. And again, we also have representatives from all the major lineages across the globe. So with that in hand, one thing that we studied very early on in the outbreak was a concept known as the phylodynamic threshold. And this, it's a concept that has been around for quite a long time, but it only became relevant for real-time analysis with this outbreak because there was so much data being generated and being made publicly available. So I should highlight that I use sometimes the term phylogenetics and phylodynamics in infectious diseases interchangeably, but they're a bit different. This plot shows along the y-axis the timescale over which epidemiological processes occur, and the x-axis is the timescale over which novel genomic variation is observed. So it's proportional to the evolutionary or mutation rate, if you will. And I've put here a few different organisms, bacteria shown in red and viruses shown in black. This is our coronavirus here. So anything that falls along this line is an organism that we can analyze using a phylodynamic framework. The premise behind this is that we expect epidemiological process to leave a trace in the genomes of these organisms, such that analyzing their genomes will tell us about the epidemiology. When the epidemiological processes occur much faster than the timescale over which they accumulate genetic changes, then we can really only apply traditional phylogenetic methods. So you can think about perhaps different parasites that might evolve slower than these microorganisms. And the idea behind the phylodynamic threshold came from this tweet by Andrew Rambo. This was sometime in late January. And he said, it looks like the novel coronavirus, at that point it wasn't called SARS-CoV-2, has reached the phylodynamic threshold. That is the point where you can estimate the evolutionary rates from the data. It will need more data before it is precise enough to be useful. And he put this, this density plot. So this is a posterior distribution that shows the evolutionary rate. Again, it's similar to the mutation rate. And the distribution is quite broad, but we see that the highest point is about 1 point something times 10 to the minus 3 substitutions per site per year. So that seems fine, but there's a lot of uncertainty. So this idea of the phylodynamic threshold is, again, when has the virus evolved enough, and when do we have enough data to estimate the evolutionary rate? When we can estimate the evolutionary rate, we can make a whole lot of other inferences that I'll talk about later. But this is a key question when analyzing these virus genome data. So how do we assess the phylodynamic threshold? Well, the first thing to consider is in phylogenetic analysis, we will typically have sequence data, we have a phylogenetic tree, and we have sampling times attached to these sequences. So let's consider this extremely simple example. We have 3 sequences, A, B, and C. A and B were collected at different points in time. And what we'll do is we've sampled these at these points, sorry. And if we measure the evolutionary distance of either of these 2 sequences to C and divide their differences in evolutionary distance to C by their time, that should give us an idea of how quickly they're evolving. That is an estimate of the evolutionary rate. This is kind of the intuition behind this. If a virus has attained its phylodynamic threshold, it means that whatever evolutionary distance we're measuring here is enough for us to estimate the evolutionary rate. And again, we also need to consider how much data we have. If we only have, say, 3 samples as we do here, then we can't really argue that we have attained the phylodynamic threshold. We just don't have enough data to estimate these things precisely. Another way to inspect this, which we actually do in practice, is we'll take a number of sequences and conduct a regression of the phylogenetic tree from the roots of the tree to each of the tips, and put that as a function of time, the sampling times. And if all goes well, this is the pattern that we expect, such that we can trace a straight line through these points. the idea behind this is that as more time has elapsed since the origin of the outbreak at the root, we should expect more molecular evolution. And this molecular evolution, if it's occurred at a roughly constant pace, should give us this sort of pattern. So this would be a data set or a virus that has attained the phylodynamic threshold. In this root-to-tip framework, the slope of the line corresponds to the mutation rate or the evolutionary rate. I'll use those terms interchangeably. And the x-intercept is the age of the root because it corresponds to a root-to-tip distance of 0. So often we look at these plots to determine whether our data have, we say that they have temporal signal, but if it's an early emergent outbreak, we would say whether it's attained its phylodynamic threshold. And in practice, this is the way it looks like for coronavirus. So this tree is just here for as an example, but this root-to-tip regression is for data collected quite, I wouldn't say late, but sometime in February. So we can trace a straight line through these points, and then we can calculate a few statistics. You could look at the R squared, which is the point, the extent to which these points deviate from this regression, and then the x-intercept and the slope, as I mentioned earlier. But what happens with an outbreak that has just emerged, where we only have maybe tens of sequences, that looks like this. If you look at these, at the plot above, these estimates here are very similar to what we have here, but if we had no idea about what these estimates should look like, it'd be quite difficult to decide whether this data set from this virus has attained the phylodynamic threshold or not. So what we do is we consider an objective method to do this, as opposed to just looking at root-to-tip regressions, which are a bit arbitrary. And the way this method works is we'll consider two models. And I should mention that this part of the talk is a little bit mathy, hopefully not too much. But yes, so we've got these two models. One model is a heterochronous model, that is where we consider the sampling times of the sequences. And the other model we refer to as an isochronous model. So we ignore the sampling times and we assume that everything was collected at some point in time, but at the same point in time. This is known as an ultrametric phylogenetic tree. And these are the two models that we'll compare under this framework. The method is known as BETS, Bayesian Evaluation of Temporal Signal. And we implemented it in a Bayesian framework because that's where most of our phylodynamics will take place. And the idea behind this is that if you've ever run a Bayesian phylogenetic method, you would probably have used a Markov chain Monte Carlo, and the goal is to estimate this quantity here, which is known as the posterior probability, and that is the probability of the tree, some parameters given the data under some model. That is equal to this term here, which is a phylogenetic likelihood multiplied by the prior and divided by this quantity here, known as the marginal likelihood. The reason why I'm bringing this up is because in most analysis that you would do, you never calculate this marginal likelihood. This is because of the way Markov chain Monte Carlo works. It has a nifty trick where we don't need to calculate this. But it's actually what we want if we're to compare models under a Bayesian framework, because it's giving us the probability over all model space. The reason why we don't calculate it is that it's actually quite tricky. This marginal likelihood, the probability of the data given the model, is the probability of the data given the tree parameters and the model multiplied by the prior. So a likelihood averaged by the prior, and we have to integrate over all parameter space and also add this over all the trees. So I don't intend anyone to memorize this. The point is that it's a very difficult thing to calculate. We cannot do this analytically in most phylogenetic contexts. So we use approximate methods, and those have been shown to perform quite well. So we implemented a few of them in this BETS framework. Once you have calculated your marginal likelihood, which you can do using BEAST1, this is where the method is implemented, although you could use this in other Bayesian programs, phylogenetic programs. But the BETS is, this model testing framework is simply the ratio between the marginal likelihoods of a model that has sampling times, a heterochronous model, and an isochronous model. So you take this ratio, and that is the score that you'll use for BETS. This is similar to, it's actually the same thing as a base factor, except in this case, the only thing that differs between the model is whether we include sampling times or not. And what we did for this project was we actually quite early on, from late January, we started collecting all the data that were available in GISAID. At this point, we couldn't really do this because we got thousands of sequences, but at the time there were only tens of sequences. So we started collecting these data maybe every week or every few days, and using this BETS framework to determine when we actually reached the phylodynamic threshold. And this is what the results look like. So I'll go through these plots a bit more slowly. The y-axis is a log-based factor, so that BETS factor that I mentioned earlier. Higher is better, so 0 is good, and the negative values would refer to lower scores, so less or lower statistical fit. Each bar represents a model, and each panel is each of these snapshots that we collected. So these are the data that we collected until January 23rd, February 2nd, February 6th, and so on. And this is the number of sequences that we had available at the time. The first two bars, actually the first three bars, correspond to a model where we include sampling times. The black one is a model where we assume that every branch in the tree has the same rate. The gray one assumes that branches can have different rates. So it's a slightly more realistic, but more complicated model, so it has more parameters. This gray one, so the next one, is a model where we include a vastly uninformative prior and some parameters. This is just to check that our prior is not affecting the way we select the models. And the other important bars here are the red ones. So these correspond to a model that has no sampling times, and then the gray ones here correspond to models where we've randomized the sampling times just as a control. So the first thing to take from this plot is that in January 23rd, the best fitting models are these models that have randomized sampling times, followed by these models that have no sampling times, and then we have the models with the correct sampling times, meaning that at this point we hadn't attained the phylodynamic threshold, because again, the best models are not those that have the correct sampling times. But by February 2nd, the picture is a bit different. The models that we select are always those that have sampling times, and that's the same situation from February 2 onwards. Note that the scales are a bit different here. And another interesting finding is that in February 24th, when we have 122 genomes, the best model selected is this one, so the gray one, which allows for some evolutionary rate variation. So this is a slightly more complicated model, as I mentioned. And what this means is that by that point, we have enough data to justify using a more complex model. Prior to that, we're using this model that assumes that the rate of evolution is constant for all branches in the tree. So more data means that we can use more complex models, but the phylodynamic threshold here was attained quite quickly by around this point. So with only 47 samples. Of course, we can do this because we've sequenced the complete genome. If we had a smaller portion of the genome, I would imagine that we would infer that the phylodynamic threshold was attained a bit later. But what are the implications of the phylodynamic threshold for our actual estimates? Fine, it was attained sometime in February, early February. Well, these plots here show the prior and the posterior distribution. It's important to inspect prior and posterior distributions in Bayesian analysis because that tells us how much the data are informing our estimates. So that's kind of just a routine check that one should always do. The y-axis are the estimates of the evolutionary rate or the time of origin of the outbreak. The light blue is the prior, so those are all the assumptions that we put into a model without considering the data. And then the dark blue is the posterior. So this is the estimate when we actually include the data. Each column corresponds to a different point in time, a different snapshot, if you will. And remember that the first snapshot that we have, the January one, had not attained the phylodynamic threshold. And in that case, what we see is that the evolutionary rate is quite low. So we tend to get very low estimates of the evolutionary rate before we attain the phylodynamic threshold. And the time of origin is also quite uncertain. In fact, the mean is, I think, mid-October 2019, which is unreasonable if you consider epidemiological evidence and how quickly the virus is evolving. But after that time, after we attain the phylodynamic threshold, which is from the second column onwards, the estimates pretty much converge to the same, to a value. So the evolutionary rate would be around 1 times 10 to the minus 3 substitutions per site per year. And the time of origin is sometime in late November, which is more in line with what we know about when the virus was first detected in China. And this had cool implications for different studies. So this is a study that Lachlan presented a few seminars ago, a few weeks ago, about direct RNA sequencing and the early evolution of SARS coronavirus. So using a lot of early collected data, including those generated by direct RNA sequencing, we presented this tree that included some of the geographic diversity available at the time. And we were able to date this tree such that the branches correspond to time. And you can see that the origin of the outbreak would have been sometime in late November, but we can also date some of these clades that were around at the time. At this point, we would probably use a different or a more wide diversity for the samples. And then this is the right plot shows the evolutionary rate estimates. The central one is for SARS coronavirus 2 using different subsets of the data, either to maximize diversity, to take the maximum number of samples, or a small number of samples, 66. And they're all quite similar, so that's quite good. But we also compared this with other closely related coronaviruses that had infected humans, in particular MERS and SARS coronavirus 1. And you can see that the estimates are quite similar. So around from 10 to the minus 4 to 10 to the minus 2 substitutions per site per year. So that is 2 orders of magnitude, but that's around the range of values that we would expect for these viruses. I should mention that these are independent estimates by other groups. So we do expect some variation depending on what methods they use and what data were available to them. And more importantly, now that we have evolutionary rate estimates for coronavirus that we think are reliable, what does this tell us about the evolution of the virus if we compare it to other viruses? This is a paper that I published with Eddie Holmes a couple of years ago. And we didn't look at coronavirus at all. We just looked at evolutionary rates for different microorganisms and how they were associated with genome size. In general, we have this, there's a lot of theory behind this pattern. So we find that evolutionary rates scale negatively with genome size. And this is actually quite interesting, but I don't have time to talk about why this happens. But the point here is microorganisms with larger genomes tend to have lower rates. So SARS coronavirus sits around here. And when you basically put it into this regression, it has a rate that is around what we expect given its genome size. And to put some context into this, this is influenza. So influenza has a smaller genome and it also has a higher rate. Sometimes it's twice as high or even more than SARS coronavirus. So slower than influenza, but around what we expect given its genome size. So nothing unusual about its evolutionary rates. And just to summarize these aspects about the phylodynamic threshold, what we find is that SARS coronavirus 2 has a similar rate to other coronaviruses. The outbreak probably originated in late November to early December, and we know this because we consider our estimates after we attain the phylodynamic threshold. And the virus has attained its phylodynamic threshold, but it should be regularly assessed for local outbreaks. So this is not to say that we can analyze any coronavirus data without conducting these tests of the phylodynamic threshold. It might be that in a local outbreak, the virus hasn't had enough time to evolve in that particular location, such that it might not have attained its phylodynamic threshold. So we need to take that into account for our analysis. And the next thing I'll talk about, which is I think more relevant at this stage, is nowcasting using phylodynamics. And nowcasting is a term that has been around quite a lot lately. So even the Prime Minister has mentioned it, and it refers to being able to infer what is going on now at present. And the reason why it's interesting to do this using phylodynamics is that phylodynamics is typically a set of methods that we would use with a retrospective focus. The reason for that is that the lag between outbreaks happening and genome data being generated used to be quite long enough such that whatever phylodynamic estimates we conducted were not relevant for current outbreaks. But the 2016 West African Ebola outbreak and the recent Zika outbreak are probably the first cases where we've been able to conduct these analyses nearly in real time. This is some of the modeling that was presented by the government and was conducted by, or was led by Jody McVernon and James McCaw. Jody's at the Doherty Institute and James McCaw is, well, here at Melbourne Uni. And this is the estimate of the effective reproductive number for different states in Australia. This is using, the data here are case counts. So these are not phonemic estimates. And in case people are not familiar with the effective reproductive number, although I doubt it, this is the average number of secondary infections that we would get from a single case. And any control measures that we put in to suppress or to control the outbreak aim at bringing the reproductive effective number below 1. Once you bring the effective reproductive number below 1, you expect the outbreak to die out if you can keep it below 1. So this is the situation in different states, and we'll focus on Victoria. We were working on these phylodynamic methods before this modeling was presented, but when it was shown to the public, it was actually quite convenient because it allowed us to benchmark our phylodynamic estimates. The aim is not necessarily to compete with this other traditional epidemiological methods. Rather, we want to see whether we can improve on some aspects that are a bit harder to capture using traditional epidemiology. I'll explain those a bit later. So what we did was we did all the, actually all the Australian data in GISAID from a few weeks ago, but we'll focus on the Victorian samples. So those are shown here in this tree in red. And the premise behind phylodynamics, or the reason why we can link evolution epidemiology, is that we know that different demographic processes lead to different phylogenetic trees. So the phylogenetic tree, its shape, its number of samples, and distribution of node times will give us information about what demographic process generated it. Here I've simulated two trees according to two different exponential processes. I've chosen exponential process, or an exponential growth process, because this is exactly what we expect to happen in an outbreak, an exponential growth in the number of cases. The tree here in red was generated under this red curve, and the tree here in black was generated under this other curve. And the way these curves are described is the number of infected individuals at some time t is proportional to the number of infected individuals when the last genome sample was collected, multiplied by e, to the power of r, which is a growth rate, but this r is different to the effective reproductive number that we saw earlier. They're related, but not quite the same, and multiplied by t. So we can fit this model to any of these trees to estimate these parameters, and the parameters of interest for us will be this, this epidemic growth rate. The epidemic growth rate is quite useful in the sense that it allows us to estimate the doubling time. So it's simply log2 divided by this epidemic growth rate will give us the doubling time. That is the time that it takes to duplicate the number of infected individuals. And we can relate this epidemic growth rate to the effective reproductive number using the following. It turns out that the growth rate is actually the infection rate minus a recovery rate, how the rate at which individuals are removed from this process. And then the effective reproductive number is simply the infection rate divided by the recovery rate. So if you do a bit of algebra, you can work out the effective reproductive number using this, this framework. And in this particular formulation, this is a coalescent simulation, or not simulation, formulation, but you can also use birth, death, or other methods for this. A key consideration here is that you need to make some prior assumption about preferably the recovery rates, and I'll talk more about that in a couple of slides. So these are the estimates that we get using two flavors of the exponential process that I mentioned earlier, the coalescent and the birth death. This is the posterior distributions for the effective reproductive number, and this is the doubling time in days. So they're quite similar, but they're telling us that the effective reproductive number would be around, I'd say 1.2 and maybe 1.5. And the doubling time might be around 15 to maybe 30 days, depends on which model you think is better. So these models make different assumptions about the sampling process and stochasticity. So I do expect these estimates to be a bit different. And here I'm making the key assumption that the recovery rate is 36.5, 1 over days. So this means this parameter is actually the inverse of the duration of infection. So I'm assuming that people are infectious for 10 days. And this was taken from the prior modeling by the Australian government, or that was reported to the Australian government. So this is, it's fine, but at the time we hadn't seen the other modeling, so it looks okay, but a fundamental problem with this model is that when we look at the phylogenetic tree, I think we have a question. Emma.
Today, I think you might mean years.
You're absolutely right. This is years. Yes, I apologize for that. Yeah, so yeah, of course, because it's 10 divided, 365 divided by 10 days. So, right, the fundamental problem with that model is when you look at the tree and look at the distribution of the samples collected, if you do a histogram of your samples, and I strongly suggest you do this whenever you attempt phylodynamic analysis, the distribution of sampling times looks like this. Here we've had a constant sampling effort, so we do expect this exponential growth in the number of samples collected, but then after this point, sometime in late March, we have a decline in the number of genome samples collected, and that matches or the fact that we had a decreasing number in detected cases. So assuming a model that only has constant exponential growth is probably wrong, which is probably why we had these RE values above 1. So what we considered is a relaxation of the constant exponential growth models. And one of them is known as the birth-death skyline. The idea behind this is that we take our phylogenetic tree and we cut it into equally sized time intervals. And within each intervals, we'll estimate the effective reproductive number for each of these. The problem with these intervals is they have to be equally spaced in the tree from the origin to the present. The default this is implemented in Beast 2, by the way, and the way this is implemented is it will give you a default of 10 intervals. When we apply this model to the Victorian data, we get this pattern. So this plot here is known as a birth-death skyline plot. The y-axis is the reproductive number, and the x-axis is time. So you can see that we have a lot of uncertainty around here. I should also mention that each of these lines is a trajectory of the reproductive number over time. And we plot these individual trajectories because this is exactly what you should get from a Bayesian analysis, a set of trajectories. So the darker colors correspond to more less uncertainty around some value. Right, so a lot of uncertainty here, but then we can detect that sometime in late March, there was a sharp decline in the number, or in the effective reproductive number. When we first had this plot out, we noticed the government modeling, and this was quite encouraging because if you look at their estimates, it only really captures this point from early March to early April. And this is similar to what we have here, except we only have 2 estimates. But it matches our finding that RE dropped below 1 sometime in late March. So that was quite good. And again, our RE estimates are quite similar to what they got. The other interesting thing that we can draw from this model is that we can estimate a sampling proportion, which we find it's quite uncertain with the GZAID data, but it's very high. So it suggests that we are making a big sampling effort as we expected. So if the sampling portion was a lot smaller, that might tell us that we have much more transmission that we're not capturing. So another way to put it is the genetic diversity of the virus could not be explained or would require a much larger number of samples to be out there, of cases to be out there. And then once we knew about the modeling that was reported to the government, we considered another model that was a bit more simplistic, but probably more informative. Sorry, because one thing to note here is that these breakpoints are extremely tight. And that is because we have a very good estimate of the origin of the outbreak. So what we considered is another flavor of the BERT that Skyline is we implemented in Beast 2. If you go to my GitHub account, you can find the code to replicate this model. And the way it works, we'll consider a single interval. So we'll calculate an effective reproductive number at the, I'll call this the early stages of the outbreak, and an effective reproductive number at the late stages of the outbreak. So only two values. But the time when this parameter changes will be a parameter in the model. So the output should be a posterior distribution for this this interval time. So the model is a bit more simple in the sense that we have fewer intervals, but we have to estimate the time of this interval, and that will be a free parameter in our model. And when we conducted that, the results were, I think, quite cool. The first thing to note is that we estimated the time of change in the effective reproductive number to be sometime in late March. And the credible interval around this is actually quite tight. So that's good. And I should say that this matches our knowledge about restrictions that were put in place and which we know have suppressed our outbreak to some extent. And then this is the effective reproductive number. Here I've just switched these histograms around. So this is the light colored histogram is the effective reproductive number at the early stages. And this is the effective reproductive number at the late stages in the darker shade. So again, if we compare that with the standard epi modeling, we have very similar effective reproductive numbers. And one thing that is potentially beneficial about these is that actually James McCall pointed this out in a recent seminar. If you note, the estimates from the most recent time points are quite uncertain. And this occurs because we have a small number of cases. So we don't have a lot of data to estimate these reproductive numbers with a lot of confidence, or with very low uncertainty. But with genome data, because we're using the branching process in addition to the samples, we can potentially make these a bit more precise. So some considerations about using phylodynamics for if we're attempting nowcasting or reconstructing epidemic outbreaks are the following. So in Australia, we have highly effective genome sampling and testing where phylodynamics works well and it's easy to benchmark. But if we had no idea what our estimates should be, I think it's important to treat these with caution. As I mentioned earlier, you need to make some assumption about, for instance, the duration of infection, or have some idea of the sampling proportion. So if we assume a different duration of infection, these estimates might vary a little bit, and our duration of infection does not necessarily apply for other parts of the world. And I also think it's important to highlight that phylodynamic estimates require substantial local knowledge. So here we do expect one point or a point in time where there was a major change in the reproductive number because of the way containment measures were put in place. So this knowledge is important to choose and configure the models that we're interested in working with. And data quality is a potential problem, especially when there are a few genomes. In Australia, we have a large number of genomes, again, from a very densely sampled outbreak, and the genomes that we have are in general of very high quality, but in some geographic locations, this is not the case. So we have to proceed with caution. For instance, this is where assessing the phylodynamic threshold might be useful. If there's a lot of sequencing errors, I would imagine that a test such as BETS would indicate that that we haven't attained the phylodynamic threshold. So conducting these tests is important. And then for the future, as travel becomes less relevant, genomics will be key to understand local transmission. And again, I think this is because we're not using just case counts, but also the information in the genomes, the branching process. So that will give us more precise estimates. Genetic structure will become more important. So at this point, we have data from the global diversity, but of course, I would expect that some clades will become more important here. So analyzing those outbreaks will be useful using phylodynamics. For instance, if you have a phylogenetic tree, it's easy to tease out whether you have two different clades spreading or a single clade spreading, and whether you should differentiate these using a phylodynamic threshold, phylodynamic framework. And at this point, I should mention that we've repeated these estimates using only travel-associated data and only local data, and the estimates are the same. Essentially, we don't have big differentiation yet. Phylodynamics will also be useful to detect super spreading events, because you can imagine these as branches in the tree that have a lot of individual branches stemming from them, so lineages with a lot of branching events. We can also estimate the proportion of undetected cases, and this would be useful if we want to get at assessing asymptomatic transmission. I think that's still a bit tentative, so we still need to work on these methods, but the models that we have at hand do allow us to estimate the proportion of undetected cases. And the way to think about this is, what we're assessing is whether the genetic diversity that we have captured, if our sampling is good enough, can be explained by the number of cases that we have reported. And we can also make these estimates for periods with no detected cases. So you can imagine a phylogenetic tree with quite sparse sampling over time. Even points in time with no samples collected will have branching events. So those can be used to estimate epidemiological parameters. And I think those are a few of the key ways in which phylodynamics can help us track our local outbreak. And there's many people to thank, but first my funding sources, the ARC, NHMRC, and the Department of Microbiology, my collaborators in Belgium and Scotland, different people at MDU that have helped me curate data, look at these estimates, put them into the right context, and think about how to model these things properly. And finally, my research group, we have weekly discussions about phylodynamic methods, and it's been incredibly useful to understand the scope of these methods and keep track of all of the research being done out there. And thank you for listening. I'll take any questions now.
Many thanks, Sebastian. That was great. So just while you're thinking of your questions, everyone, could you please just put a question in the chat? And I mean, you can just indicate briefly what the question is in the chat, and then I'll call on people in turn to raise the question. It's about 10 to 12 now, so we've got quite a few minutes for questions. We have recorded this, and Sebastian just needs to check with co-authors about whether we can make the recording available, but we hope that will be possible on the MIG website. And for the same reason, Sebastian's very kindly shared with us really kind of, hot off the press research. So, don't communicate anything without permission. Can I just jump in and ask a question first while others are thinking? Sebastian, when you gave us those first estimates of the evolutionary rate and the origin time, yes, the very first one, which was before the phylodynamic threshold, it was kind of curious that the origin posterior was very diffuse, which you'd expect, but the evolutionary rate posterior was highly concentrated on what we now think is the wrong value. So that shouldn't happen, right?
Yeah. So what I would have expected is the evolutionary rate, the posterior, to match the prior. Right? Is that what you mean?
Yeah, similar to the prior. I mean, that's true for the time of origin, right? The posterior is not too far off the prior. That's what I would have expected for the evolutionary rate as well.
Yeah, well, I've seen this happening many times, and I think what happens is that you still have some genetic diversity, only a handful of mutations. It's pushing the root to be, I mean, the prior is, I don't know, I think it pushes it to be a bit old, and that means that the rate tends to hit the zero boundary. I think that's why this occurs.
Okay, thanks for that. So Simon Firestone, do you want to just unmute and ask your question?
Yeah, sure. Can you hear me okay?
Yes.
Yeah, wonderful. Wonderful. I've got one word there.
Sorry, we are now losing you. Maybe, can you just try one more time? Otherwise, I'll read out the question.
Is that better?
Yeah, that's good. Yeah.
Wonderful. Yeah, great talk, Sebastian. I just wanted to ask what you think the priorities now are at this point in the outbreak, given there's a lot of focus now on following clusters and checking for otherwise unknown chains of transmission out there.
I think we need to look at, well, this is my take on it, right? I think we need to, we have to expect that we'll have, local transmission will be more important than travel, of course, but once we open borders, travel will be something too. to monitor constantly. For localized outbreaks, I think we do need to think about improving, I guess through contact tracing you can do some of this, but any undetected transmission, for instance, if we think we have an epidemiological cluster and that doesn't match what the genomics is telling us, we do need to look into that in a lot more detail. What I'm saying is we might see a few cases coming from a household. We do need to nail down whether they all come from the same source or not, because that might tell us about underlying community transmission. So I think that's where we need to focus this. And of course, ultimately that will tell us about undetected or asymptomatic transmission in the community.
Thanks.